All right, so I'm going to finish up the macromolecule workshop that we started. So you should have some of these down. Um, so I might go through some of them a little quicker than others. Okay, so first, remember the difference between a monomer and polymer. So you should have this down, but if you need to stop and write anything else down, go ahead and do so. But remember, mono means one, poly means many. So if we start off first with proteins, remember the building blocks are amino acids, and that the main elements that are found in proteins are carbon, hydrogen, oxygen, and nitrogen. And nitrogen is that unique element that's only found in proteins. And some of the main foods are eggs, nuts, beans, meats, vegetables, and tofu. And that there are 52 amino acids, and that 20 of those are essential to our diet. And so we need those 20 in our diet to make all the proteins that we need in our body. And 9 of those amino acids aren't made in our body, so we have to eat them to be able to get those 9 amino acids. So if you think about how amino acids are linked together to create proteins, you can look here. This is the monomer here, so one amino acid. And then to create the polymer, amino acids are linked together like a train here almost. And this blue line in between each amino acid is the bond. So we've talked about covalent bonds and ionic bonds and hydrogen bonds, but this bond is unique to proteins, and so it's called a peptide bond. And these peptide bonds are very important to the structure of proteins and for those amino acids to be linked together to form those proteins. And so one thing to remember with proteins is that the structure equals the function. It's very, very important that you remember that, because if this process here does not work properly, or if the folding after this that makes a more complex functional protein doesn't occur properly, then that resulting protein is not going to work properly, which is then going to cause problems for, other things within the body. So one type of protein that's especially prevalent within our bodies are enzymes. So enzymes are proteins. That's a big thing to remember. And like one of your vocab words, enzyme, these enzymes are going to speed up chemical reactions in your body. So chemical reactions are occurring all the time in your body, even as far as you breathing in oxygen and breathing out carbon dioxide. That's a chemical reaction. So, but these enzymes, for instance, are going to be the enzymes that help to break down the food that you eat. So the first place that starts to happen is in your mouth with amylase, which breaks down sugars in your mouth. And so it does it pretty rapidly. And so, like if you were to chew on a saltine cracker for an extensive period of time, and it wasn't a salted saltine cracker, it was just a plain saltine cracker, you would notice that the taste would go from cracker to almost sweet. And that's because the amylase is breaking down the sugars, the complex sugars, into simple sugars. And so there are a couple of things that can disrupt that functioning, and that's heat or pH. And so heat, like I talked about in the workshop, was from really high fevers, especially in babies. That can be very dangerous to degrade that protein structure, and also pH. So our bodies have specific pHs throughout it. So there's a pH for your stomach around 3. There's a pH for your blood around 7. So it's important that our body maintains that normal pH throughout it. And so then if we look at enzymes specifically, and remembering that those speed up chemical reactions, there's a model that is used to describe how enzymes function, and that's called the lock and key model. So this model is used to describe the functioning of an enzyme with its substrate, another vocab word. So the substrate is what binds to an enzyme and changes its shape to speed up a chemical reaction. Okay, so if we look at the pictures, it's a 3-step process. So first you start with an enzyme and a substrate that are not together. And that substrate is then going to bind to a specific active site on that enzyme. So it's like your key to your house hopefully only unlocks your house. So if you go to your neighbor's house and try to get in their house with your key, it won't work, and vice versa. So the substrate can only bind to one specific enzyme's active site. It can't bind to another one. So amylase, for instance, has one specific substrate that binds to that to speed up the reaction of breaking down sugars. So after that happens, then it changes. It has a complex, so an enzyme substrate complex, which then speeds up the chemical reaction, and then the enzyme is altered, and then the substrate is released. And so this process is pretty complex, but that's the simple model of what's used to describe how the enzyme and substrate kind of work together. Okay, so if we go to carbohydrates next, the carbohydrates building blocks are sugars and starches. So a lot of times carbohydrates, people think, oh, potatoes, um, you know, fruits, candy, all that kind of stuff. And so those are all carbohydrates, but there are different kinds of carbohydrates. There are simple and complex carbohydrates. So the simple ones are going to be called monosaccharides. So those are going to be your glucose, fructose, sucrose, etc. So sucrose being like table sugar, fructose, high fructose, and simple forms of glucose in candy and chocolate and things like that. So those monosaccharides then can be linked together to create polysaccharides. So the polysaccharides are going to be complex sugars like your starches, cellulose, and glycogen. And starches, starchy foods like pasta, potatoes, bread. You think about if you boil pasta on the stove and it's a starchy thing, obviously, that you get this white foamy stuff on the top. And that's actually the starch bubbling out of the pasta. And especially happens if you boil potatoes. If you ever make mashed potatoes from scratch, you get tons of white foamy stuff on the top. And so these polysaccharides are the ones that are more beneficial to our bodies, because these simple sugars, if you eat a candy bar, it's going to spike your blood sugar up. So it might give you energy right in the moment, but then you're going to crash because it doesn't have anywhere to, take you. It just does it very rapidly. It rapidly raises that blood sugar, and then you just crash right back down. But these polysaccharides are complex, so it takes more to break them down. And so you kind of think about people that are athletes or cross-country runners especially, and that's why they kind of carb load before they have an athletic event. So then their liver can store that glycogen and then break it down through a process called gluconeogenesis, and their body can then use that glucose. So those polysaccharides are much more beneficial for long-term storage. So then, if you remember from one of the Plickers questions, whether our bodies could break down cellulose, these structures below kind of show you why we can't. So cellulose here is just a linear chain, and our bodies are not able to break down that linear pattern, and so we can't utilize it like cows can because they have multiple stomachs to break down the cellulose and actually use that energy in their cells. But we break down the starch, which has this branched pattern, and we're able to then break this bond off and actually use that starch. Okay, so then if we go to lipids, our building blocks for lipids are fatty acids, and remember there are two kinds of fatty acids, saturated and unsaturated. So the saturated kind is the unhealthy kind. So it's saturated by hydrogens, and it's typically solid at room temperature. So you think about things like butter, whole milk, peanut butter, vegetable oil, margarine, etc. And then we have unsaturated, which is healthier, and it has less hydrogen molecules, and it's usually liquid at room temperature. So things like this would be avocado, olive oil, fish oil, flax, and canola oil. So the main thing to remember with these is that they're really just long chains of carbons that have hydrogens attached to them. And depending on if they're saturated or unsaturated, they have more or less hydrogens. So if you think of the word saturated, something saturated, it's completely surrounded with hydrogens in this case. So once again, foods would be margarines, oils, omega-3s, butter, avocado. Another place that lipids are found are in our cell membrane. So we'll talk about this more when we get to cells, but for now, know that there's something called a phospholipid in our cell membrane.